What that means, you could use this graph on if you number of the engines and around be implemented at that level, and also people could be. So we have done this of JQ, and with that.
Center is tech.
It's called forest rest of the ocean. Percent of many threats, but climate change, ocean acidification, you can see it has attracted biodiversity, fish, and it's really colorful. Now, what happens when you bring in all these threats, including pollution and all this? So your corals will tend to bleach. So bleaching means they tend to lose the symbiotic relationship between microorganisms. And an interesting fact is that not all of them bleach. Some are resistant to the bleaching events, some bleach and die off, some bleach and bounce back to life. So that happens when your corals bleach. So you have a degraded reef, and you'll end up losing all this. So what is being done to ensure that we don't lose marine biodiversity? So we have communities coming up to sort of conserve, like they form community-led organizations to try and protect the marine resources. We also have government setting up marine protected areas, but that is not enough. We're still having challenges, including climate change. So a slight increase in temperature will lead to bleaching and corals will die. So we have conservation organizations now coming up with methods of trying to restore the corals. So for example, they go into the ocean, pick up fragments that have been broken off their reefs and try to grow them in nurseries. Okay? So you can even see this nursery here is attracting fish and other marine organisms. So after that, they go try and outplant in the degraded reefs. Okay? So if you look at this, you need to ask yourself a few questions. Now, there are more than 6,000 species of corals that have been described, but this classification is based only on morphological characterization, which is not very accurate. So we are trying to help them to know what species they are working with. So because we'll find some species are hybridizing and producing different species that are very difficult to understand, like to classify using morphological patterns. And the next question is, how diverse are these species being restored? And what's the best outplanting pattern to reduce effects of inbreeding and outbreeding? Okay? So because inbreeding can lead to loss of fitness, resilient genotypes can be lost, and all these things. So we are trying to bring in science and help this conservation organization to try and do a sustainable restoration of coral reefs. Now, to do this, we have sampled coral reefs that are mostly in the areas of restoration and done DNA isolation from them. So the next thing is to try and come up with a library for RADSEC sequencing. So considering the genome size of corals, about 300 MBs, it's too large to say you want to sequence the whole genome. So you need to come up with a strategy to help you do something called a reduced representation genome sequencing, where you just target a specific section of the genome and do analysis on that. And that will help us come up with diversity and relatedness analysis. The second thing is to try and do a PCR, like amplify a mitochondrial region within this species and try to come up with taxonomical annotations from them. So when all is said and done, we hope to try and answer all the questions that we raised initially, to try and come up with an inventory of all the species based on molecular information, understand relatedness, and help these people try to do a sustainable restoration of corals. Thank you.
Thank you, Rodney. You've kept time. Now we are on to the second last presentation, which will be given by Stephen Kasioki. Stephen, welcome. Please let's clap for Stephen as he comes to the podium.
Thank you so much for the opportunity. As the ICT department reflects my presentation, I introduce myself. I'm Stephen Kasioki. I'm an MSc student in the Institute of Zoology, Chinese Academy of Sciences. I'll be giving a review on the importance of soil mites, or rather Oribatid mites, and the status of their diversity globally and locally. Soil Oribatid mites, they are majorly commonly known as beetle mites, or the moss mites. Another name is amad mites. And I've realized that since yesterday, we've never had a talk on below ground biodiversity, and that's my area of focus. And allow me to just kindly briefly, because of time, go through my review. Yes, as I've said, this group of arthropods, they are majorly the most diverse and numerically dominant, occurring in the soil profiles and the litter. They are also found in leaves, barks of plants, and they are very tiny. Actually, their size for an adult one ranges from 0.5 to 1.5 millimeters. So you have to use a microscope to see them, unlike the larger animals. They are primarily terrestrial, but only around 90 species of all described species of the Oribatid mites. 9 species are aquatic. They occur in large densities, and you can find almost 100 to several thousands in individuals in a cubic meter. Their global diversity, not quite much have been done on them, and as you can see from the map, this is their distribution globally. Like in the areas of Neolithic, there's over 1,100 species have been described. In the Palearctic, there are over 3,000, and that's where much of has been done. You come to Ethiopian regions, where we are now, Africa. We have over 1,400 species have been described. Come to neotropical regions, over 1,800. The Australian is over 1,300. And just generally, globally, over 11,000 species have been described by different researchers. This number reflects a general over 1,300 with 163 families globally. Now we come to or rebatted mite research is in Kenya. And according to a review and a checklist that I prepared, I found that not so much research has been done on this group in our country. Only a few have been described, and I found that only 109 species, representing 8 genera and 40 families, have been described in Kenya. What does this tell you? That there is need of much research to be done in our country and also in Africa to understand the biological diversity of these insects and also the taxonomies of these insects. And these are just areas where this group has been described in Kenya. As you can see from the map, with Nakuru, Nairobi, Safo, and areas of Shimba Hills, Mombasa, Mount Kenya, Tana River, and Malindi. So there's quite a lot that we need to be done as scientists. Sorry? Yes, what the ecological role of these mites? I know a lot have been done, said about the importance of insects, the importance of different ecosystems. And one is they are very vital in decomposition of organic matter, the litter content of all that. And also they are very also important in nutrient cycling, as they help in bring down, or rather recycle the nutrients and make them available to plants. They are also good in soil formation and structure, improving the soil fertility through their burrowing activities in the soil as they mix this, and also improve the aeration of all this. And they are also good indicators of ecosystem health and the functions. And what are the threats? I know there some have been said, and indeed, each and every organism is facing threats, and majorly, man has been put in the focal point as the main driver and the cause to all the threats mentioned, from habitat and destruction to pollution to issues with climate. Man plays a key role. So as scientists, there is need for us to take control of what the activities that we do to our ecosystems. And just an estimate on the rate of extinction of this group. It is fairly estimated that by 2030, if there are no actions taken, we might be losing from 0.6% to 6.0%. So are we going to face mass extinctions as we sit and watch, or are we going to address interventions to control our ecosystems? Just a conclusion of this, the research of these mites is very crucial into our country and to the global world because of the importance of this group, from decomposition, nutrient cycling, to the monitoring and addressing issues with ecological health and functions, and also the implication. There is much need and urgent research needs to be done, and particularly into our country, to address issues of taxonomic updates of this group, and also issues with conservation efforts that need to be addressed, because limited studies, there are limited studies in our countries to just address the complications of this taxa. And also, currently, I'm engaging on a research on their biodiversity in one of the biodiversity hotspots regions in Taita Hills. And I'm comparing the diversity in different ecological systems, the natural forest, the agricultural, and the plantation establishment, the exotic plantations, to know how is their trends in species richness, abundance, and all that. So with that, thank you so much.
A hearty clap for Stephen. Okay, now, last but not least, we have the final presenter. I think we are doing well. We are just about to conclude the session. David Onyimbi. Again, David is taking us back to the coast.
Thank you for this opportunity. I hope you guys are having a great afternoon, and try and take us to lunch as fast as I can. So I'm Onyimbi David, MSc student, University of Nairobi, and I'm working on an endangered species, the Hildigardis tombat, Taphozoas hildigardiae. Taphozoas is an endemic to the coast of East Africa. It's only found in Tanzania and Kenya, and in the islands of Zanzibar and Pemba. It was recently updated by the IUCN in 2020 as an endangered species from near threatened status. This is because its numbers have been declining rapidly, and the habitats have also been greatly fragmenting. So this species is a cave-dwelling species. It only lives in caves, and some of the reasons why cave-dwelling birds prefer caves are permanency, protection from predators, and microclimatic conditions that are stable due to their high sensitivity in osmotic properties. That's my bird squad there. This is the species, Taphozoas. The male species usually has a black beard, as you can see there. That's a key identification of the male species of this bird. So it's actually easy to identify them when in the caves. Problem statement. The major reason we are doing this study is because there's a lack of ecological understanding of the roosting ecology of this bird. It seems to only select specific caves within its range. There can be literally a cave at the walking distance to the next cave, but it will select only a specific cave. So this is what we're trying to understand in this study, why it is selecting these specific caves, and why the numbers are declining so that we can curb it from there. So the justification of the study is to help identify and protect critical habitats to the species, guide effective conservation strategies. Of course, this cannot be started without understanding the ecology of the bird, and avail knowledge vital for urban planning around the habitat range, because there's a lot of development going around the caves, and some of them tend to be destroyed, especially in the coast where there's a lot of tourism, and also enhancing monitoring and management of the bird populations. General objectives is to determine the influence of cave microclimate, physical characteristics, and macrohabitat heterogeneity of the tombat. Specific objectives are broken down as follows. So we are going to critically examine the microclimatic characteristics of the caves. We are going to examine the physical characteristics of caves, and we're also going to describe macrohabitat surroundings of the caves by GIS techniques. This will help us try to understand what this bird prefers and what it doesn't prefer. So we're going to do a comparative study where we're going to select target caves which the species lives in, and proximate caves which the species does not use. And then we're going to compare all these characteristics and get to understand why it prefers certain caves to others. So our study area, mostly we're going to sample Kilifi County and Kwale County at Shimoni, Kilifi County at Watamu. These are some of the areas in the coast with a lot of coral caves that are used by birds. So in our methodology, we're going to deploy data loggers in the caves, and these data loggers will be programmed to record microclimate, characteristics of temperature and humidity every hour of 24-hour days for 7 days for each cave. Then we're going to compare these among the caves. Those are some pictures of the equipment. At the far end is a data logger that records temperature and humidity readings. And this one is an envirometer used for wind speeds and also temperature and humidity readings in snapshots. There's installation of data loggers going on in the caves. So we try to install the data loggers as high as possible, close to the roosts of the birds, so that they can capture the data accurately for the roosting of the birds. We're also going to assess physical attributes of the caves by doing actual physical measurements. And when doing physical measurements in cave environments, you want to disturb the birds as little as possible, so we won't be using conventional tape measures and all that. We'll be using a lesser distance measure. This enables you to measure a cave as quickly as possible and minimize disturbance on the species. That's a picture of us working on physical attributes in one of the caves. For physical attributes, we're also going to try and quantify disturbance in these caves. The caves are actually quite disturbed by human activities around them, and we're going to have to try and promote sustainable practices in the activities that are happening around there to ensure that we are not interfering with the birds. So the first picture is at NMK Shimoni, where we have the Shimoni Slave Cave. Shimoni Slave Cave is one of the habitats of these species, but apparently whoever is running the cave weren't aware that they have an endangered species in the cave, so we had to highlight that issue to them. And tourism there is one of the major reasons why we're having a lot of the species migrating from that cave to nearby caves, and especially because they have to install lights, white lights in the caves so that tourists can see, and these lights interfere with the birds when they're roosting during the day. Some caves in Shimoni, in the villages of Fikirini, are really exploited for guano. That's the middle picture. That's guano exploitation that happens at night. So when they're doing this, they're really interfering with the habitat of these species. So we're also trying to sensitize a lot in the community about sustainable guano harvesting. The last bit of the study will include macrohabitat description, where we're going to involve a lot of GIS techniques in categorizing the land use change and land use description of the areas to try and see if there's a link between how the land is being used and interference with the habitat species, the species of this habitat. So this study is at its preliminary stages. We are currently actively collecting data, so I won't have any results to present for you at this moment. But when we actually decode all the data from our loggers and the rest of the objectives, we're going to do some analysis and maybe in the next opportunity have to present to you. So we expect the study to provide light on cave-dwelling bird species by identifying specific parameters affecting its behavior. And future studies would also use this information as baseline. That will mark the end of my presentation. These are some of the partners, both financial and technical, that we're working with to ensure that we save this bird. Thank you.
Thank you very much, David. Okay, I think we've done very well. We lost a bit of time, but we have already forgiven ourselves. And I'm going to beg your indulgence. Do we take questions because there's provision for 1 minute plenary, or is lunch calling too loudly? Is there any burning question, 1 or 2 or 3? That's all we can allow. Okay, there's 1, 2, 3. And that's it. 3 questions. Please address the specific presenter.
Thank you. My name is Orero Eric. I'm very, very much interested with my friend who has presented about the part. I will wish to see your mitigation measures. And if you are interested in much of the parts, just go to Kisii on your way to Kisii prison. There is a menace of parts, thousands of thousands. Please give us the strategy on how to control the parts in terms of noise and everything in that area. Thank you.
Question number 2.
Thank you so much. So my name is George. For those who were here yesterday, you will understand I speak for my grandmother. So I'll start with Joshua. These are very simple, but it's all related even to the second presenter. Because I want to make a statement and say there seems to be a disconnect between research and reality on the ground. Let's even start from the bat conservation, because I happen to have an understanding of all the areas that he's talking about where these bats are. The understanding of the community, including myself and my grandmother, is that bats are a problem. When they get to the houses, at least the species that live within the houses, they are a problem. And if you look at the research that we are doing, there is no connection between the real reasons for conservation and how that connects to communities or people in terms of then how they benefit from that conservation. We are doing research, we are getting degrees, but where are we leaving our people? So how does my grandmother understand why they need to conserve those? For what? And so the same applies to when you're talking about, for example, the macro invertebrates that are indicators of water health, maybe the safety of the water. So how does a person, because some of those stressors that Joshua was talking about are local. But how does the person on the ground understand? How and when do we cascade this so that then it becomes part of what you call the citizen science? That people can themselves sample and understand where they are. I think we are doing research for degrees, but we're not doing research for development. Because unless we connect it to the lives of the people, I don't think that this research is being a lot useful. Because again, most researchers are in town. Our grandmothers are in the village where all these problems happen.
Okay, the last and not least question.
Thank you. Thank you. I had a question for Irene. Remember on the flycatcher, but I'll ask it later. Also a question for Benjamin. I'll ask later. My specific question now, if I have only one, is to Joshua and the bats. This is a critically endangered bat that only occurs really in coastal areas. Correct? Yes. You haven't said what it actually eats. Is this a fruit bat, insect bat? Does it suck blood? And what may be, let's say, its specific foods that restricts its distribution to the coastal area?
Okay.
Can I answer my question? I think my question was asked earlier on.
Okay, proceed.
I hope I can remember well. Did you ask me how many species of stonechats are there in the world? Did you ask me how many species of stonechats there are in the world? What was the question?
Bird that you're studying. Bird that I study. Well aware. It's actually the same species that also occurs in Europe.
So they're all subspecies, actually.
They're all different subspecies.
Yes, they're all different subspecies.
But if it occurs from the equator all the way to Northern Europe,
Yeah.
yes, if you would also study the European populations, it would be a good possibility to prove whether the Bergman rule actually applies.
Yes.
If you stay in Kenya,
Yeah.
yes, maybe your gradient of temperature is
Yeah.
not big enough to recognize the effect of temperature on body size.
Ah, okay, okay, I understand now. Oh, thank you.
Okay. Thank you very much. I think there is still time for us to meet in the corridors and to discuss. We cannot be able to answer all the questions in this session because
I'll answer quickly. All their questions are interrelated, and I love that. So I'll start with yours. Sorry, I didn't mention the species and what it eats and all that because of time. There's a lot about bats that I can't demystify here, but it's insectivorous species. It feeds on insects, the locust family and the butterfly family, and it also feeds on mosquitoes. Okay? No. But the bat only occurs on the coast. So that's why we are not examining what it eats, because those occur everywhere. We're examining other parameters, yeah? We good? On that note, I go back to your question in Kisii. So you're saying there are a lot of bats there being a menace. And back to his question, the species, there are different bats, insect eating, fruit eating, nectivorous, blood sucking. In Kisii, there's definitely a reason they're there. Could be that they're eating fruits in Kisii, or nectar, and they're very important for seed dispersal, pollination, and all that, okay? So if you remove them there, first of all, you might not be able to continue having those fruits in Kisii. So it's not a task of just going and removing the bats and we're done, okay? So there's a lot of things that go into control of the menace. There are solutions for that. There is a technology called bat houses. It's not yet common in Kenya or Africa. It's mostly used in UK, where there's a lot of protection on bats. So there are actually bat houses that are developed through roost ecology. If you understand roost ecology, you'd be able to develop a very nice bat house that would be specific for the species that is a menace in your community. So if the species in your community is studied, roost ecology, alafu designiwe bat houses ziekwe hapo, zitatoka kwenye ziko, and they will go there. It's happening in UK. You can Google it and see more about the bat house technology. For my friend there, the last question, I love your concern. You've expressed a very nice concern, and it cuts across a lot of research going on. A lot of research is just being done for degrees, but I'll speak for myself. My research is a microfacet of the work going on in conservation of the Hildigari storm bat. It started before I even thought of bats, and I wish you would get a chance to go to the coast, Shimoni especially, and visit the Three Sister Cave Complex. There's a lot of work going on by BCI, Bat Conservation International, where they're trying to help communities living with bats create sustainable livelihoods around the bat ecosystem. So they're actually training people to know how to identify these bats, protect these bats, and leverage on these in tourism. They're actually even buying land pieces where these caves are that are owned by landowners who might go into development, and creating conservancies that are employing people there. So in the coast right now, a lot of people are very happy about bat conservation going on, and they're very interested.
Thank you very much.
We'll
Thank
talk
you
in the
very
corridors.
much. The chair has
We'll
the
talk in the
mic.
corridor.
The chair has the mic. Thank you. Okay, for this session, we have had mentorship for students. We have also had research scientists very well trained and ahead in career. And we hope that the students have learned something, and we also hope that the early career and the middle career scientists have also found areas where they could guide the students so that research becomes more and more relevant to Wanjiku. Now, lunch has been served. Lunch is ready, but before you go, I would like you to put your hands together for all the presenters, for the audience, and for the IT team. Thank you very much. Thank you very much. Lunch is served in our usual, the foyer. Please, let's find our way there. And the next session begins at 2. Yeah, it's beginning. Thank you. Okay, please take the least amount of time that you can because you are the audience and the presenters for the next session. Thank you.
Fauna, and I'm calling upon Dr. Jane Macharia. But before she comes, maybe I tell you my name. My name is Eric Mudama from AKE, Association of Kenyan Entomologists. Thank you. Welcome, Dr. Jane.
Good afternoon, everyone. Yeah, as you have heard, I'm Jane Macharia, and I'm going to start my presentation. Yeah, earlier speakers spoke about species, but I would like to blend the species and ecosystem. And I'm working on wetland ecosystem, the agricultural activities and the impact it's having on the biodiversity, and whether we can come up with a middle ground. Wetlands are becoming the refuge now that rain-fed agriculture is becoming more and more rare. So we have seen influx of people doing agricultural activities in wetlands. And wetlands have important ecosystem services. As you can see, provisioning, regulatory, cultural, and supporting. But within here, we also have biodiversity. My focus is on biodiversity, which again, this time being insects, they also offer all these provisioning, like the bees and the honey, regulatory, cultural. There are some cultural association with insects and supporting services like the nutrient cycling and all that. Yeah. Okay, meant to go. Yeah. So, what do we want to do? Do analysis of agricultural wetlands interaction. We do research. Then we see the driving indicators for sustainable wetland use. Then we compare decision for the outcome. Then we can recommend. And as I was saying, human beings are the major drivers, and then the pressure, then the state, the balance state of the ecosystem is affected, hence the ecosystem services, then we have to respond as researchers from our findings. So, again, these are some of the activities, the opening up of wetland, the agriculture, the chemical use, the overgrazing, over-abstraction, vegetation harvesting, and all these will have impact on biodiversity, including invertebrates. So, what was the problem? The problem is land transformation, leading to decline in productivity and a threat to biodiversity. Then we need determinant of such wetland sensitivity and vulnerability to anthropogenic activities that are largely unknown. Then biodiversity lacks even the most basic data on most species-rich group, especially invertebrates. A lot of studies have focused on the big, maybe the big five, the big mammals and such. Again, also, small wetlands like the highland floodplain, that is Ewaso Narok. A lot has been done on the major lakes, like Lake Naivasha, Lake Victoria, but the smaller wetlands that are important as they are the ones, they are living with the communities. So what was the research question? How does land use change affect occurrence, abundance, composition, and diversity of beetles? This is the species we are exploring for development of indicator species, with the sampling dates along a gradient of flooding regime. And then, is there potential for beetles as indicator species for anthropogenically impacted wetland? Then, this is the study area. Ewaso Narok is in Rumuruti. And the flooding regimes I'm talking about, this zone rarely floods. This is seasonally floods. This one is permanently flooded, and activities, agricultural activities do not always happen. Then this also is seasonally flooded. These are the climatic of the area. The lowest rainfall is in February. The wettest month is July. Then the warmest is March, and the coldest is August. Yeah, what did we do? We did the sampling, as I said, in the crop, where there were crops. The crops is where they were doing the cultivation of mostly tomatoes, maize for subsistence, but tomatoes were done almost at a horticultural scale, commercial. Then they abandoned some plots. They moved to other places. Then the grazing, and then the Palestine areas. And I said, this is the land use gradient, and the flooding regimes, and the seasons. Yeah, so we did pitfall for the ground-dwelling beetles, and the sweep for the canopy-dwelling beetles. We had a total of 42 sampling per each sampling season. So these were the findings. For the ground-dwelling beetles, we had this species in the dry and the wet season, and the sweeps, and this is the total. So in total, we had 17 families, 143 species of beetles were recorded. Then how was the diversity? Along the land uses and the flooding regimes, we found that there was no significant difference between the diversity, both in the land uses and also in seasons. Same for the canopy-dwelling beetles. Then what about the composition? Here, we see that the composition along the land uses were more, there was like the sharing of the species, but here that there were some land uses that were not captured here as far as the composition was concerned. So again, the composition and the land uses and the other parameters like the flooding regime were not significant as far as the composition of the beetle species were concerned. These were for pitfalls, and it was the same. We tried to use this, the PCO, to see the sampling dates. Like October 2016 and October 2015, there was an overlap of some of the beetle species that we found. But in the July, not so much, July 2015 and 2016, and same with November, there was a distinct between in the composition of the species. Same with the seasonality, and these were the land uses and the flooding regime. These were the sweeps. Again, here we have the composition of during the wet and the dry season, and the flooding and the land uses. But there are some cases where with some of the land uses were not captured. So the composition was not as significant even in the sweeps. Here the sampling dates, July 2015 and July 2016, we see here the July 2015 was different from the July 2016. It was the composition, you can see the distinction here. And we were trying to see that the way the seasons, they are no longer the classical dry and wet season. Times have changed, and the years had different rain. I mean, the millimeters of rainfall were different. And then in the land uses, there were some species that were only unique. These are some of the species that were they only occurred in the cropland, and this only occurred in the fallow land, and this occurred only on the grazing land, and here in the pristine. Maybe without this maybe could be the ones that can help us develop indicator species for the different land uses regime. So we are going to explore further on this. So what, in the discussion and conclusion, we got 143 species from 17 families. It's a good representation of the known beetle families. 2, the findings have added to the knowledge of beetle occurrence and distribution. And the beetle families also belonged to various functional groups, thus providing the ecosystem services we talked about, like the family Chrysomeridae, occurring mostly in the cropland. And some of these species and the functional group could be used for biological control, not forgetting the other ecosystem services like the helping in the decomposition and nutrient cycling. Then Ewasonarok is being utilized a lot for agricultural activities, and we need to develop indicator species that can be used for land use planning to strike a balance on agriculture and conservation. Then here, however, the species that were unique, both in season and in the land uses regime, were basically less than 5 species. So while maybe they may not be enough to develop indicator species, they could be used going forward to form a baseline to continue with the monitoring and probably see what indicator species can be used to define the land use regimes. This work was done 2015 to 2016, and there are so many changes that have happened. So one of the recommendations would be maybe to continue with the monitoring to see what could come out of it. Yeah, I would want to thank the GLOBE Wetlands in East African program that was supported from the University of Bonn and the National Museums of Kenya, and the expertise that helped in the identification and field work. I would want to acknowledge one person who is here, Maurice Mutua. Thank you so much for the identification. Thank you for your audience. Thank you.
Thank you, Dr. Jen, for the very powerful presentations. We could agree how best to clap for each presenter. Either we do many small ones or one big one. Okay? Or you can clap the Korean style. They like this. Right? We'll agree. Now, you have noticed that on the program, you are given 10 minutes to present. I was lenient kidogo for Jen. Dr. Jen was the part of the secretariat organized for this conference. She was the chair of all communications. So all the emails you got and communication was from her. So based on that, we gave her some 2 more minutes to finish her work. So kindly observe the 10 minutes time given. We'd like to hear from Mr. John Kipyegon, kindly, for the next presentations.
Good afternoon, everyone. My name is Maurice Mutua. I'm standing in for Dr. John Kipyegon, the one we did the work together with Dr. Esther Kioko. So we are here to present our results, the findings from the title, Enhancing Resilience of the Sacred Mijikenda Forests. And our case study was based on Kayakauma. Kayakauma is one of the 7 UNESCO sites listed under the World Heritage Sites. And these forests, they are under the management of the Council of Elders and also the National Museums of Kenya. For the Council of the Elders, they manage the forest using the traditional beliefs and practices. That's why, as NMK, or National Museums of Kenya, we are coming in to introduce the scientific view of it. And this is what I'm presenting. Our main objective was to understand or establish the composition and abundance and also the distribution of invertebrates and fish across the forests, forest edge, farmlands, and also the river. And also our mandate was to identify some of the ecosystem services which can be derived from the invertebrates which occur in Kayakauma and the surrounding areas. The study materials are there, and as I've mentioned, the study was carried across different habitats, which is the pristine forests, at the forest edge, farmlands, and also the river, because we wanted to understand the disturbance gradient of this ecosystem. Some of the methods which we utilized were pitfall traps, pond traps, baited butterfly traps, time-limited searches, and also aquatic nets for the aquatic macroinvertebrates and also for the fishes. Data analysis, the data which was obtained was analyzed using software Prima 5. And for the water quality, we used an index which was improvised and devised by South Africans, which is the South African scoring system, which gives an index based on the level of tolerance for invertebrates. Level 0 to 5, those are the ones which are less tolerant and they can't tolerate pollution. So in areas where you get those species, it means the water is clean for human consumption and also for animal consumption. For cluster 2, which is 6 to 10, that is moderate. And above 10, it means those are highly tolerant. So wherever you get group 2 and 3, it means that water is not that safe. So some treatment is required. About the results, as I've mentioned, our study was more on a disturbance gradient. As you know, in farmlands, there are several activities which happen there. At the edge of the forest, people have access to the edges of the forest, but given that this is a shrine and they operate, they manage them based on traditional beliefs and practices, most of the people they fear entering the forest. So the results reflected and corresponded to those beliefs, because what we obtained, the forested areas, it had the higher diversity. Although the abundance is higher in the farmlands, the diversity, as shown in this table, you can see the diversity. It's very high in the forest, and it's followed by the forest edge to the farmland. These are the results from the terrestrial, not from the aquatic, because the methodology are different. We analyze the results different for the terrestrial and for the aquatic macroinvertebrates and fishes. So the results are clearly depicted. You can see 4.4, 1 for the forest, 4.3 forest edge and very low in the farmlands. But when you go back to the abundance, you realize the farmland had higher abundance, followed by the river, the forest area, and lastly the forest edge. Going to the species richness, the forested area had the higher species richness, followed by farmland, the forest edge, and the least was the River Nzofuni. Now, and that is well depicted in the tables, in the graphs which are there. Another interesting thing is a new record of Abitu, which we collected from the forest area. It's a Casida, which is yet to be described, but it's a new record for Kenya. It's a species which has never been recorded again, and scientists are working on that to establish its identity. So that shows one of the importance of these shrines or the kayas. Now, we also tried to compare the species overlap, whether there was a clear separation of the different species which we recorded, and our question was well answered by some of the analysis. As you can see, S3, all the points, we had 3 points for site 3. You can see they have clustered together, and there is no S1 or S2 within the clustering of S3, same to site 1 and site 2. You can see the sites are clumping together, the forested area, the forest edge, and the farmland. So there was a clear separation of species distribution amongst the different sampling areas. The same happened with the stream. As you can see, upstream is a region which occurred within the forest edge. The river was in between the forested area for the upstream. For the downstream, that was within the farmlands and also the human settlement. And you can see there is also a clear separation. There is no overlap among us, the different species which were recorded for the two. About how effective was our work, and if we exhausted all the species which occurs in Kayakauma. Our species accumulation curve reveals that more needs to be done. A lot of sampling effort needs to be employed because the curve is still expanding. So more work is required based on the analysis as per the species accumulation curve. For those index which I've just mentioned, when it comes to upstream, you can see this is the upstream, this is downstream. I mentioned about the categories based on the SAS, that is the South African system of scoring species. You can see group 1, which is most tolerant to pollution. You can see there are several, the bigger percentage was of the most tolerant species. The smaller percentage was of the moderately tolerant. There is no group 1, which is least tolerant. So that sends a message which I'll communicate to you as we proceed, and the same was replicated in the downstream. Those are some of the species and the way they were grouped based on their response to pollution. Now, we also identify some of the ecosystem services based on the functionality of the groups, and it's interesting because what we got, pollinators were the most abundant, and you know the roles they play in our ecosystem. Predators, you understand the role they play through controlling of pests, and also pests which are also dangerous, but also decomposers were very high. So with the decomposers or the nutrient recyclers, the pollinators, and predators, it means that area is safe because most of the species which support most of these functionalities of the ecosystem are more abundant than the one with the negative effect. Under the discussion and conclusion, we documented 415 species, and this was after a study, a baseline study which was carried in 2019 by myself and Dr. Esther Kioko. During that time, we documented 251 species, and during the COVID-19, which is in 2020, additional 100 plus species were added to the checklist. So you can see currently the species of invertebrates and fish which have been documented for that area are 415. Also, the recording of higher species or the higher diversity of the forests, it suggests that the forest is still an important area because it domiciles important invertebrates which requires to be conserved. Another thing, the absence of the tolerant species in the aquatic system, it shows that something needs to be done. There must be some human impacts, negative ones, which are impacting on the ecosystem, and something needs to be done to rectify that and to reintroduce those candidates which have been lost. Also, the presence of those moderate tolerant species to pollution in the upstream, it suggests that the water is of moderate quality and it needs to be treated before human consumption. Also, there was a recording of most of the edible insects and other crustaceans, including prawns, crabs, and fish. So this calls for some aquaculture practices within that region. And for the discovery of the new record, it means the forest domiciles other species which are of importance, and more studies need to be carried out to establish those other species which are in the forests and not yet documented. Some of the recommendations were that more ecological surveys need to be conducted. As you borrow from the species area curve, you realize the species is still growing. It hasn't leveled. So more studies are recommended. For those who are looking for proposal, you can suggest Kayakauma as one of your areas of study and get to establish some of other species which are in the forests. We also, a lot of awareness is required for the locals and also training for them so that they can derive benefits which are domiciled in those ecosystems, one of which is aquaculture and also promoting insect-based enterprises. We do thank the African World Heritage Fund for funding the project, also the National Museums of Kenya for granting time for us to conduct the research, and also the local community, volunteers, and also the Council of Elders of the Kaya Forest for granting us access to the shrine. Thank you.
Thank you, Maurice, for the work done by Rajon. You've told us about sustainable work and also the new species. You haven't been very keen on time, and I think that is not very sustainable given the remaining 14 of them. So we need to be more sustainable with time. Kindly. Now, our next presenter is a member of staff at NMK. I'm glad to invite Dr. James Odanga for his next presentations. So kindly, Daktari.
My name is Odanga. Title of my presentation is Spatial Distribution of Oriental fruit flies in smallholder avocado orchards along altitudinal gradient of Taita Hills. My co-presenters are Professor Olbayo, Professor Nyakanga of Nairobi University, Dr. Samira and Dr. Ekesi of ISIPE. Next. Avocado is an important crop in Kenya, and Kenya is the major avocado producing country in Africa. It's very important people to know. So Kenya produces a very huge number of avocado. For the export market, it's about 19.04 billion in value, Kenya shillings. All these avocados, including what is eaten here locally, are mainly produced, 70% are mainly produced by smallholder farmers. And these smallholder farmers generate good income and nutritious food for themselves, apart from for the country. However, these farmers face challenges, many challenges. Key among them is insect pests. Among the key insect pests that may invest avocado are false-called moth. This is mainly a pest of immature avocado fruits. Common blossom thrips, this is mainly a pest of avocado flowers. Greenhouse thrips, it's mainly avocado young fruitlets and leaves. Scale insect is a new emerging pest of avocado currently, and oriental fruit fly. Oriental fruit fly is a serious, invasive, exotic quarantine pest. And it is polyphagous, means it affects many crops and many wild fruits also. In terms of our research, it's mainly a post-harvest pest. It attacks mainly avocado when avocado fruits are mature. And for information, avocado fruits mature only when they're harvested. Avocado fruits only ripen when they're harvested. They don't ripen when they're not harvested. So conversely, it is a very poor host. Avocado is a very poor host for this fruit when the fruits are still on tree. That means they're not harvested. Our specific objective were, 1, assess percentage damage level in mature avocado fruits by B. dorsalis along altitudinal gradient of Taita Hills. 2, describe the population density of B. dorsalis along Taita Hills. And the third one was about to describe how this population, what changes, what make this population to change. This is the map of study areas I worked. The red line from around here up to people who have been to Taita Hills. Taita Hills, for people who don't know, it is in southeastern Kenya. It's about 100 kilometers, 150 kilometers from the Indian Ocean, and about 90 kilometers from Mount Kilimanjaro. So we designed this area into 3 altitudinal zones: the lowland, the midland, and highland. And the elevation gradient between where I worked is between 900 and 1800. So along these gradients, we measured the infestation level of mature avocado fruits during harvesting season. We measured weather variables using digital machines, and we also measured the population. We assessed the population and infestation. For temperatures and weather, we had automated weather stations along these stations. All along this, temperatures was measured every 30 minutes for 2 years. Rainfall, we had weather stations, which were temporary, and humidity and other parameters which are measured. So one of our major results was now to see what infestation of B. dorsalis on avocado fruits. The yellow ones represent fallen avocados which were collected under the tree in farmlands, which are fallen. That means they are ripened or they are ripening process. So they are more infested in lowland. The avocados which are collected in lowland areas were more infested, followed by the midland, and in the highlands for the fallen avocado fruits was very minimal. For on-tree avocado, because B. dorsalis doesn't mean that it doesn't attack, because on-tree it can also be, and harvest fruits can also be attacked by birds, can be eaten by small mammals, so they can have injuries. Those injuries, the female B. dorsalis can lay eggs, and when you go to the lab, you might get one or two getting into a mature adult or a larvae. But at the highlands, it was near zero. So the infestations at highlands, for both, it was almost near zero. It means that the highlands above 1500 is an important area that farmers can do good avocado farming without near threat of this pest. For mean number of B. dorsalis, similarly, the lowlands had a very high abundance, and that is very normal for B. dorsalis. It is registered as a lowland loving insect. It's also an exothermic insect that it depends on temperature, high temperatures, to reproduce, to fly, to do all things that a live insect needs to do. In the highlands, it was almost none. Where we got one or two specimens was near the market or high schools or church or clinics, what we call market centers mostly. And interestingly, when you correlate the infestation level, lowland, midland, highland, for both fallen and on-tree, and their burdens, it was possibly correlated at a very highly correlated. This is just a show of some weather patterns of weather parameters we collected, just a representative temperature, mean rainfall. So temperature was high in the lowland and low in highland. Similar, conversely, in the highlands, rainfall was high, and the lowland was, yeah. So we used these parameters. We collected these parameters because one reason was to define our ecological or agroecological zones or altitudinal zones. And one of the key things is weather parameters. They're really defined, and they defined they're very independent. And we use them, including other parameters, now to evaluate how what influences distribution of these population of B. dorsalis along latitudinal gradient from 900 to 1800. And we used, all these analyses were done in R, statistical software. Here, to model this, we used linear mixed effect models. And the linear mixed effect models, we designed models based on the parameters we collected. And it was found that, so for the models, to evaluate the models, to choose the best model, we use models. We use AIC, which is called Archaic Information Criterion. And AIC, the one model that has the lowest AIC, is the best model. And for our case, using all those models, the model that was best was one that had temperature only. So temperature influenced best the population of B. dorsalis along latitudinal gradient of tighter hills. Yeah, in our conclusion, it is important to note that avocados mostly are planted in the central region of Kenya. And this central region in Kenya, mostly they're planted above 1500. So it's one of the safest places. So any area above 1500, avocado is a B. dorsalis is not a major problem. It is a major problem after harvesting. So the only way farmers should focus on this is to do what we call cleaning up of, there's a word they use, but the easiest way is cleaning up of the fallen avocado fruits. Every day, every few days, collecting and preparing them in a standard way. CPA has standard ways of how to collect and keep them somewhere so that they don't reproduce. And one of the things that we noticed that if you want to control this pest in the farmlands is also that we start earlier before the population grow up. Key impact of our study that after 2018, that's when we published a paper you're seeing this, there was a lot of hype and acceptance of avocado farming, what you see in the country now. Interestingly, at the same time that the lifting of phytosanitary, which was there, our AKE chairman talked about it, 2008, the lifting of that was there from 2008 up to 2018, that's when it was lifted, the phytosanitary export, to export avocado to other countries. And the data, weather data sets we collected contributes to precise climate information, the current climate information of Taita Hills. Thank you.
Good. Can we give Dr. another clap, kindly? He's been very gracious with time. He has saved us 1 minute. Thank you very much. Now, we did agree in the beginning that we need to note down our questions, which you could probably ask. I'm noting down my questions. So kindly, if you present, we're able to have a session of the question and answer session. I'd like to also call our next presenter. His name is Timothy Mwinami, if I got that correct, to also present his work. Thank you.
Yes, thank you. Good afternoon, everyone. My name is Timothy Mwinami. I'm going to present my work on movement and distribution of lesser flamingo in East Africa. Okay. Lesser flamingo are iconic species in the Rift Valley lakes. They are one of the unique species lake indicators on this alkaline lake within the Rift Valley lakes. And they are unique because of several factors. From the taxonomic, lesser flamingo is unique out of the world 6 species of flamingo, which belongs into 3 genus. But lesser flamingo belongs in its own unique genus, Phainoconius, and is nearly threatened despite the fact being the most numerous and abundance in large number. It occurs in a very large number within the East Africa Rift Valley lakes. East Africa represents or holds 75% of lesser flamingo globally. And it plays a very major role in terms of foraging and feeding of lesser flamingo. Why is lesser flamingo unique? From the ecological component factors that support its survival, up to its specialized food, which is blue-green algae or Spirulina fusiformis, it remains a very unique species. And as you can see within these 3 sites, this is the microscopic food or stable food for lesser flamingo, which is Spirulina fusiformis. But of recent, the food is changing gradually. As we go along the presentation, you'll see. How is the species distributed globally? Lesser flamingo, as you can see, it is found within North Africa, East Africa, South Africa, and India. That is the global population. Our study was based in Kenya, and then on Lake Bogoria National Reserve, or Lake Bogoria Soda Lake. Within the global population, we have 2 populations. We have one population that holds, or we have 2 sites. One site that holds the breeding population, and another site that holds only the foraging population. The breeding sites globally are only 5. In Botswana, India, Namibia, South Africa, Tanzania, Natron, Madikadikadi, Etosha Dam, and Sewage Dam in India. What are the threats that is affecting this species to be categorized as nearly threatened? We have climate pattern, which is changing the aquatic system, the food density, and the food quality for the species. We have socioeconomic aspect, eutrophication, encroachment, and pollution, which is changing the wetland. And then we have regular mass mortality. Once the food changes on the lake, the birds can develop into some diseases or weakness, and then they die in very large number that the records are not well followed. What was the objective of this study? After the long rain season in 2012, all Rift Valley lakes, everybody will agree with me that they rose up, and the water quality changed a lot within the Rift Valley lakes. So this eliminated to the species distribution into different areas for their foraging or for survival, which made this study to be developed or for us to carry out this study. And we wanted to identify regular routes for the species in order to give advice to policymakers for this aviation people, for socio-economic development like mass, wind powers, to avoid the routes to reduce the mortality or collision. The second reason why to identify the refugee sites. When the lake level has increased, where are the birds? These wetlands are not yet marked as flamingo sites. So we wanted to identify these sites for conservation purpose. Then to identify threats and document them per site. You will agree with me that all wetlands have got different threats and they thrive in different aspects. So you cannot say that all wetlands have similar threats. So identifying threats on Lake Bogoria, Lake Nakuru, Elementaita, Makati, Wasonyiro, and Natron is very key for the conservation of lesser flamingo. Methods used. For trapping the tracking species, we use 2 methods, mist net and pie chart. Pie chart is a wire mesh with fishing line noose on it, which can assist trapping, and is mostly used in Western countries for those species that are within the artificial dams. But for the wild population, we used mist net, and the main reason was to assess the effective, cheap, and faster method for trapping the species. And then for tracking, we used solar and plastic rings. The reason for using these 2 methods was to assess the cheap, long-lasting, and the most that can be used to generate a lot of data for long-term monitoring. Then for assessing the food, it was collecting water samples from the lake, bringing them after fixing it with a chemical or formalin certain percentage, bringing it back to the lab to assess the food density. Then for monitoring purpose, we used vantage point to assess the population within specific points on Lake Bogoria. Our results, we captured 9 lesser flamingo in September 2022, February 2023, 30 individuals, and then 2024 we did not capture any. Population estimate, we had 158 number, 59,000 and 190,000. What does this tell you? The population has drastically reduced within these lakes because Lake Bogoria rarely recorded less than 300 historically trend, and you'll see as my presentation ends. Then, because of the cost, I had only one microsatellite GPS that I was to tag one individual to see how the movement will happen within this one individual. So I tagged one individual after assessing the weight, the most healthy one, which was 2 kg, and I tagged with the metal ring of D4903 plastic ring BB. From our estimation, we discovered that each site of the lake hosts a certain population, whatever time you go there. If you want to see a lot of lesser flamingo, if you go to the hot spring, if you go to the hot spring, you'll find the largest population. And if you are looking, maybe you have tourists or you want to study, you're looking for a small population, you can visit maybe the Old Cat or Loboi Inlet River, and you'll find this population. For 3 assessments, we found this population, these sites maintain certain number of flamingo that cannot exceed. And this explains the ecological condition of the lake, how it supports the food density for the species. Then, this can explain also very well from the open waters. Open water, you'll see during our first assessment, it had very low numbers. Then the second assessment, but the third assessment, all the birds were in open water, or most of our birds were in open water. This explains the foraging behavior of the species. From the now tracking the individuals, I haven't received the GPS coordinate, but I've received the map, which are here. You'll see in one single day, lesser flamingo can make several movements. If this map was complete from the communication I had, I'm still receiving the data. It reached Elementaita and then it went back the same day. So what does this tell you? This is a very mobile species which moves very fast from one site to another, be it daytime or nighttime. Initially, we understood that lesser flamingo are nocturnal migrants, but later on, we've known that lesser flamingo migrate both time, during daytime and during nighttime. Then from the population assessment, you cannot specifically say that Lake Bogoria, neither Elementaita, neither Nakuru has got constant population. They change a lot depending to the lake quality, water quality, and ecological condition within the site. Because when we went in 2022, the population was almost 200,000. When we went in 2023 February, the population was less than 100. Then also in 2024, the population was almost 120. This is a very nomadic bird, depending to the situation on the site. Then from the second map, that is the movement of same individual that has got solar-powered GPS. What does it explain? It explains that the species is very nomadic in search of food. There are two theories. Initially, they said the food is all about, they smell their food, but recently the other literature is saying that they send scouts. And then the scouts theory can buy logic because, as you can see, this is a species in one day moving from one site to another, from one site to another. Searching, we don't know. Discussion. Lesser flamingo population trend is declining, and it's a fact. But to where? Nobody knows. And who is responsible for this? Nobody can respond to this. What is the way forward? As I put the end to of my discussion is that the most approachable way is to build capacity to community members who can support in monitoring, then enhance technology, incorporate technology with scientists in order to monitor the population of lesser flamingo within East Africa. We formed a group called Lesser Flamingo, Friends of Lesser Flamingo, and we've started training community members all along. Anybody who wants to join Lesser Flamingo group, you are welcome. We are giving back the power to community members to do the monitoring. Thank you so much. This was National Geographic funded project, and I thank National Museum of Kenya and all stakeholders, WARTI, Baringo County, and Kenya Wildlife Service. Thank you.
Good. Thank you, Timothy, and your team for the good work. Now, it's getting hot in here. Maybe you shake your neighbor's hand, make sure he's not sleeping. Try to wake him up. Try to see that he's alive. And then, before I call the next presenter, who is Patrick Malonza, I want to read the name of the next 5. In this way, the IT team will be able to put their presentations in order of their presentations. So, after Patrick Malonza, we'll have Justus Ojong. We'll have Aziza Zuhura. We'll have Philemon Langat. We'll have Florence Masharia. Then we'll have Michelle Lawing to mark the first 10. Now, Timothy told us a very nice thing, very interesting. I learned today that flamingos feed on cyanobacteria. I had no idea. And I know you're learning much as well. So, since you're all awake, thank you. Let's have Patrick Malonza doing his presentation. Then we'll be able to call the next 5. Please welcome. With a clap, kindly. He has a bit of work to stretch. Thank you. Thank you, the IT team, for putting the PowerPoint. So, Dr. Karibu sana. You'll have 10 minutes.
Yeah, good afternoon. My name is there. I'm Patrick Malonza, one of the pathologists here at the National Museums of Kenya, studying snakes and other reptiles, and also studying frogs and other amphibians. So mine will not be hard science kind of research. I'll just give you things on more of a challenge to people who work at the museum, especially with the specimens, and just look at how museum can contribute to the taxonomy and also promotion or even identification of key biodiversity areas. And here, as you know, museum collection all over the world, they are like libraries. So you are able to even generate maps, even write guidebooks, purely using only the collection, be it a flora or fauna kind of collection. And one key thing is that for that collection to have any value, it has to be of good condition. So, and then again, purely using the collection, you can also identify areas which are important for biodiversity, because you know the distribution of those species, and then you can map out and know which area it is. And one key thing is that what we call key biodiversity areas, in many cases, they also occur within what we call the global biodiversity hotspots. And another very important thing with the museum collection is that a museum collection is important depending on the number of collection it houses, either how many millions and all that. But one more important one is how many type specimens, be it a holotype or is it paratypes? Does it host or does it harbor in that collection museum? So maybe you can have a very big museum with a lot of specimens, but what is more important is the type material which it is. For instance, this is one of the new species from Arabuko-Sokoke forest, which is a well-studied forest, but you always get new stuff from even well-studied kind of areas in Kenya. So this is just to give you an overview of the number of species we have in Kenya, about 300 reptiles and about maybe 100 species of amphibians. And these are some of the new species. This is a Sicilian from Endau Hill in Kitui. This is a thread snake from Savo. This is a forest lizard from Kitui Hills. This is a blind snake from Beere, which is in Embu County. This is also a legless lizard from Arabuko-Sokoke. This is from Central Highlands of Kenya, and this is it's also in a new genus. It's a forest toad from Mount Kenya. And this is just a list of the species we have described since 2014. So it's in total we have got about 22 species. They range from lizards, snakes, frogs, Sicilians, and from different parts of the country, from Abadeas, coastal forests, then from even Savo, from Nyambene Hills in central Kenya, Mount Kenya, then the Matthews Range, which is in Samburu, and also from the deserts of Marsabit. We also described Arok Agama from northern in Marsabit. Then still we continue, we also describe, these are forest geckos, most of them from different parts of the country. And they just continue. These are also frogs from Nyambene Hills. And then up to just recently, this one just coming out recently, the ones from Arabuko-Sokoke, it's almost out because it has already been just for the print. And as I said, you can also use the museum collection, that is what we have described, to identify key biodiversity areas, or you can also promote others which are known, but now you can add, because in key biodiversity areas, initially they used to use birds only, but nowadays, like in Kenya, they are now expanded to other species of flora and fauna. And some of the ones which have been supported, they are like the eels in Kitui, which have got 2 endemic species. Also like in the Tharaka, the Shakarika Hill or the Kijege, we have 1 species which is endemic from recorded in that hill. Then Mount Kenya, we have got 3 species. The Kirimiri forest is in Embu, 2 species, but some of the species may occur in the nearby like the one in Runyenjes, one occurs also in Mount Kenya, because it's not far. Then we have in Laikipia, we have, among those described, 2 of them are endemic to that place, but one occurs outside the Laikipia also. In Chulius, we have got 1, which occurs only in Chulius. Matthews Range, we have got 2 species. And in Nyambene, we have got 3, but most of those 3, at least one is endemic to Nyambene. The other 2 are also found in the nearby Mount Kenya. In Sagala, we have only 1. Sagala is part of Titaius, 1 species endemic to that place. Then Arabuko, Sogo, 2 of them, which are endemic to Arabuko alone. So these are some of the, you can see also when we are doing the identification, the bellies also look different. These and these are 2 different species. You can see the extent of the color, but on the back, they all look alike. This is the same. So there are some of the challenges, as I said. One is misidentification, because if you are not sure of what you are ordering, it's better to leave it as maybe either at genus level, because when you misidentify, you also kind of, no one will come and look at it, because you've already given it a name, unless somebody is working on that group. So the same case applies to this legless lizard from Arabuko sokoke. For me, I found one which was identified as a snake, a red snake, in 2001, I think. And then it was not in good condition, but at least I was able to remove it from being a snake to being a legless lizard. And then it was only after the following year an expert came, and then he told me, this is a new species, just leave it and wait until you get specimens. So we managed to get specimens in 2009, and now the species has just been described. So this one has talked about it, about the condition. So when you just collect a specimen, don't just collect for the sake, collect to ensure that the characters which can be used to describe that specimen, at least they are all covered. So that because you don't bring a specimen which is not in good condition, so it will be very hard, and maybe it will be a single specimen. Then the other thing is, another challenge is about funds, which is just, as you know, taxonomy hardly finds, as compared to other applied biology funding. And the other thing is about equipment. As you can see, this lengless, this is the head, and then you have to use an equipment to magnify the head. That is a digital microscope, and then take that, and then you can count the scales on the head and all that. But most of these things, these equipment are also not available, or they can be very expensive. And basically with that, I think that is the end of my review, and thanks a lot.
Thank you, very senior scientist here, Dr. Malonza, for the very precise presentations. I've seen, I was reading your abstract, and I've seen that most of the reptiles you described are from where I come from. So I'll follow up to know why. Thank you very much. Now, we began this session at 14:35. It's now around 3:45. We have had our first 5 in around an hour and 15 minutes. I believe that's good enough. It's a good trend. If we manage that, we'll be able to transact the remaining part in a good way. So, well done. Well done, our first pair of presenters. So please, let's give them a clap. That was quite a pool of scientists. Well done. I'm amazed. Now, we get to hear from our next 5. We'll be having Justus Ochong, Aziza, Zuhura, Philemon Langat, Florence Masharia, and Michelle Lawing, in that order. So we begin our first with Justus. He's on his way to the stage. Justus will give you a clap as you bounce to your stage. Please come. Thank you, and enjoy the rest of the sessions. Santi.
Thank you. My name is Justus Ochong. Together in this survey, we worked with Dr. Beryl Buang, and Dr. Beatrice Hayota. I'm going to present to you on apetofauna of Ngong Hills, Kenya, Kajiado County. So introduction, rising settlement in Ngong Hills ecosystem poses a great threat to its biodiversity. This is because Ngong Hills is a source of our rivers like the Bagadi River, and also it also has some religious aspect whereby most of the churches usually use the source of the rivers for baptism. But biodiversity survey calls for comprehensive audit of the species, composition, and the distribution of the hepatofauna of the Ngong Hills. However, little is been known and is been known about the amphibians and the reptiles of the Ngong Hills. So we need to do more work for us to know the different and the distribution of the Ngong Hills hepatofauna. Our aim was to conduct a comprehensive audit of the hepatofauna of the Ngong Hills system so as to formulate the conservation strategy for the threatened species. This is because, like some of the species, like the Plimpton cacostanum, was found almost 20 years ago, but we didn't find any. Materials and methods. Our study area is Ngong Hills. It has an altitude of between 2,000 to 2,500 above the sea level. It's located in Kajiado County at that altitude of south of 01 degrees 25 minutes and easting of 036 to 38 minutes. It's about 25 kilometers southwest of Nairobi City. Climate of the Ngong Hills varies from semi-arid to semi-humid. It has bimodal rainfall from March to May and October to November. Vegetation is open grassland with patches of bushes and short forest, but it also has, like what the Kenya KFS are doing, they have some plantation of eucalyptus in those patches. Ngong Hills has got 3 blocks. It has the Kibiko block, it has Ololua block, and the Ngong Hills itself. So these are the different vegetation type of the Ngong Hills and the different habitat of the hepatofauna of the Ngong Hills. It has the forest, it has where we have the rocks and part of it are vegetation. So methodology used in our study, we did some desktop literature review from the collection of from the National Museums of Kenya and the database, and also from the field guides. These are the books which have been generated by people who has done the identification and description of the species. The field work duration was 35 days from February 2019, which was a dry season, and September 2019, which was also a dry season. But on May and January, on May, it was a wet season, and January 2022 was also a dry season. So our methodology used, we did some TLS. By that we mean is time-limited searches, whereby 2 people searching for reptiles you set your time. Let's say you set your start time, and then as you work, you say you'll be working for, let's say, 10 minutes or so. depending on the number of people you have. And then you stop, you record what you've seen, and then you start again your time. So as you can see, our hours was divided into blocks of 30 minutes each. And then we did have some pitfalls. This is a set of 5 buckets of 5 liters each, set in a plus manner or an X manner with a drift fence. And then we did our analysis performed in Microsoft Excel. So, but the difference and the pitfalls, it has to stay there for some time. This is because it's been used to take the most nocturnal animal or those who are hard to get during the daytime. So that's the drift fence. And then you also have some, these are riverine system. This is whereby, this is also part of the Ngong Hills where is the source of river Mbagathi. So from our results, a total of 154 specimens were recorded, both reptiles and amphibians, of which 22, the next one was 22 species, 6 amphibians, and 12 reptiles. Varies from different families, genuses to species level. These are our results. On survey 1, this blue number, the blue color, represents the amphibians. And which color is this? Huh? Yeah? Is it brick red? Yeah? Which color is this? Yeah? This one. Yeah? Which color is this? Red. Okay. For the red one, the red color represents the reptiles. As you can see, on survey 1, we did have fewer amphibians compared to the reptiles. But on survey 2, we had lesser, but the reptiles were much higher. When you compare to survey 3, the reptiles were also few, but the amphibians were few, but the reptiles were also higher. The same to survey 4, which means the species richness in Ngong Hills, most of them were the reptiles, but the amphibians were very few. So, and also, all the amphibians and reptiles observed and recorded were done using the TLS, apart from only few, which we got on the pitfalls. So these are different species we got from the Ngong Hills. That's a three-horned chameleon. That's a Trachylepis. And then we have the frog here. These are Hemisas, Marmoratas, and then is a snout burrowing frog. And then the other one is a sand frog, Marsabit sand frog. So on the next survey, we had 18 species, of which 6 were amphibians and 12 were the reptiles. So we also added a new records of 3 amphibians and 4 reptiles. So the amphibians we added, just to mention but a few, these are the Dantish, the Hyperorius granticola, and the Ametia witei, which is a river frog. And then for the reptiles, we also added, but to mention a few, we have the Moglas, Suduvali, which is a writhing skink, and the Crotopeltis otombeia, which is a snake. And then for the 3 species of the amphibians recorded previously were not found. We didn't find any during our survey. And these were the Cassina and Peter's nest form, and Plimpton's caco. So the threats of the Ngong Hills, we have grazing. There's a lot of grazing is being done on the Ngong Hills. And then we have illegal logging and also debarking of the tree. This is because they are using the barks of the tree for medicinal value. There was other threats there was a lot of dumping on the Ngong Hills, but more so those sides of Ololua and Kibiko. So in our discussion, as I've talked to you earlier, a total of 154 amphibians and reptiles were recorded, but there was no much difference in species richness in the 4 surveys. This is because, we recorded 13 species recorded on survey 1 and 2, which was conducted in the wet season, compared to the 10 and 12 species which were collected on dry season. Contrary to expectation, as the data would suggest, I hope you know that during wet season, people may think that you'll get a lot of amphibians compared to the reptiles, but that didn't happen to us. Instead, we recorded more reptiles than the amphibians. And then more reptiles were recorded. No, more reptile species were recorded in the wet season compared to lowest. But in the dry season, we didn't, it was vice versa. But the high abundance of both the reptiles and amphibians were recorded in wet season. The survey added new records of amphibians and reptiles to the Ngong Hills. And the current number of species we found were 27, of which around 15 were amphibians and the rest were reptiles. 2 species were recovered after close to 30 years. But no ICN list species was found. But we managed to get the species under CITES, which are the 2 chameleons, the Van Hornels and the 3-horned Jackson chameleon. So, but there also, most of the amphibians and reptiles, they're being treated by overgrazing, illegal logging. This is because in the logging, they log the indigenous tree, which is a good habitat for the reptiles and amphibians. Yes? Yeah, more or less. In conclusion, the information on hepatofauna of Ngong Hills is incomplete and calls for more studies. This is because the fate of Cassina, Senegal Cassina and Peter's nest form needs to be established. This is because these are the species that were found a long time ago in the Ngong Hills, but now they are not, we didn't find any. The study is also under threat from encroachment from agriculture and infrastructure development and overgrazing. This is because of the wind power those wind power generating, and the infrastructure like the SGR, and also people are encroaching into the forest. Thank you. So I would like to acknowledge the Government of Kenya, National Museums of Kenya, NRF, and the Kenya Forest Service. Thank you.
I have good news and bad news. I'll begin with the bad ones. Just as you have not done justice in timekeeping, that's the bad news. You have eaten to our 7 minutes. So I don't want to be another bearer of bad news. Thank you. For the good news, I just got some good news from Grace when she walked in, which I'll be telling you later about the good news, about a very interesting thing happening today. So I'll be mean for now, but let's do some science. Allow me to welcome on stage our next presenter. Her name is Aziza Zuhura, to take us through the next session. Welcome, Zuhura. You understand, thank you.
Good afternoon. My name is Aziza Zuhura. I'm from Mammalogy section. Today I'll discuss about mammals of Ngong Hills Forest ecosystem and socio-economic significance. I was working with my partner, who was Lilian Awino, and it was seconded by Dr. Beatrice Hayota. With the introduction, we have about 412 species of mammals in Kenya, and we got about 40 species found around Ngong Hills. This is a comprehensive biodiversity of Ngong Hills ecosystem that is essential because of extinction, because of the extinction of the species that are concerned. The forest currently is facing a serious anthropogenic and environmental challenges, as the cases of the forest logging is forest logging and soil erosion is apparent. Yeah, I hope you all know about the facts about the mammals. Is that mammals are the only animals that possess true hair, and females have mammalian glands and produce milk and feed young ones, and have 2 sets of teeth in lifetime, except the elephant, whose molars are replaced 5 times. Okay, did you know that shrews are one of the smallest mammals in the world? That ranges at only 2 inches long and can weigh less than an insect. Okay, the aim of this survey is that we wanted to assess the current diversity of distribution of mammals species in Ngong Hills, Ololua, and Kibiko Forest. And that was the survey of the area. We have red-pointed places that we have Ngong Hills. We have, on the map, you'll find that we have Ngong Hills, which is green, on the greener part. Yeah, we have Ngong Hills, Ololua, and Ngong Hills, Ololua, and Kibiko on this map. Yeah, that was the study survey. Yeah, it's showing now. Yeah. Kibiko Forest, Ololua Forest, and Ngong Hills Forest. That's where the research was done. Materials and methods that we used in the field to work on these mammals are, we used the snap traps of a size of 2.2, and we used the live traps and the snap traps. the shaman traps. The shaman traps are the live traps, and the snap traps are to make specimens if you want to prepare weight collections. So we place them at 5 meters each. We used to study to catch the tiny mammals. Traps were individually baited. We have to baited them first with a single raw peanut of ground nuts, while shamans were baited with oats and flakes. The traps were examined once in the morning, every day in the morning the next day, every day in the morning at 8 a.m. to 9 a.m., and then rebaited later in the afternoon. At the original locations. At the original locations, we used to leave them for 2 nights, and then we shift them to other places. And we use the same localities, or we shift them to a different location, like from Ololua to Kibiko. Yeah, so a total of number, we used a total of number of 100 snap traps and 60 shaman traps to work on this, to trap these specimens. And after every 2 nights, we change them. Then the 3rd night, the following day, we change the location. Like now, we move them from Ololua and to Kibiko or Ngong Forest. So if you check on this table, we have number of, this is Ngong Hills column. We have Ololua and Kibiko. So these are snaps that, and these shaman traps, and these are snap traps. So at Ngong Hills, we had put 35, and then Ololua 33, and then Kibiko 33. So all these are snap traps, number of snap traps. And then we have shaman traps that, these were 20 at Ngong Hills, and then Ololua 20, and then Kibiko 20. So this goes to 20 to 60 traps, and these 100 traps. So all these are the total of number of traps that we had put them down. So these are the specimens that we trapped at Ngong Hills, Ololua, and Kibiko. So I didn't put so much because of time. Yeah, so because of time. So those are varieties of species, number of specimens that we had trapped, and we also had different types of primates. So we have results of this, of the species richness. We have the Hyracodeas. So for me, I just decided to put in all of them at the same time so that to work on time. So you have like the Hyracodeas at Ngong Hills, Ololua Forest, and Kibiko, then primates, Rodentia, shrews, and then that is Ngong Hills, Ololua and the Forest, Logomorphus, carnivores. You find that carnivores, we just only found 3 of them in Ngong, and then Ololua, 2, and then Kibiko 0. That was nothing, because Kibiro, it has been tapered by, there is a lot of population. That's why we never found any of the carnivores, like leopards. So that is difficult to get them, even the Logomorphus. And the Logomorphus like hares, because people feed on them. And you find that the Satiodactylas, this is also, it's a threat on the other side. Okay, so these are now the results abundance in the forest. You find that the ones that I labeled in red shows that these are species that are highly collected, what we collected, highly collected. Those were the primates and the rodents. So at Ngong, we collected 36 and these 45 rodents, and then Ololua, 28, 55, Kibiko, 23, 57. So we find those are the highest population that we collected along the Ngong, in all those places, in those 3 forests. And this is the lowest, that's the Satiodactylas, because of there is a problem with the community that they feed on them. The Satiodactylas are like the dicks. So those are the Madokua kakis. And we have like, in this part where we have the Logomorphus, those are the hares, forest hares. So this is also, communities also, it has a threat in the community that people are feeding on them, and also the carnivorous. Some of the carnivorous, these people, they kill them, like now the leopards, because they find that it's a threat to wildlife, to what they are keeping at home, at their places. So for my discussion, for the discussions that our analysis of the results present that a research is provoking scenario, implying that with more researches, there is still more mammals could be encountered. And the failure to locate more species of mammals is not an assertion that they are completely missing. The time for searches was very limited because we were supposed to do more research, but time was not on our side. And it was not possible to visit all the probable habitats for the species. So you find that species like the bats, it was hard to work on the species of bat because there was a problem with the weather. It was too windy, like Ngong Hills, it was too windy, and we also had Ngong Hills was too windy, and we also had we also had the wind turbines that they affected the bats. They were not able to fly at the same time. So or they were supposed to fly, and they were not supposed to fly on time because they were finding difficulties. So you find like Ngong Hills Forest was noted to be exiting at a higher altitude, above 22 meters above the sea level than the type of locality on the Ololua Forest, which lies below 2,000 meters above the sea level. If high altitude specific vegetation sampled is a forest contributing to the general low number of species, it is therefore necessary to sample the other parts of the forest, which are relatively similar altitude of the type of Ololua Forest. The challenges that we faced in both Forest Ololua, Kibiko, and Ngong Hills, difficult in terrains, adverse weather, which I've just discussed about it, with the wind turbines and the community because some communities that have encroached they have built inside the forest. So wet season, or about the wet season, but visibility in the mornings was hampered by mist. Yeah, so this also affected the collection of specimens. This also affected the number of specimens that we were collecting because at wet season we collected less specimens, while on the dry season we collected more specimens. So traps were captured, interfered with by herders. Okay. What I'll recommend is, in conclusion, the findings of the rapid survey underscored the importance of proactive conservation, measures to safeguard the rich mammalian biodiversity for Ngong Hills, Ololua Forest, and Kibiko in the face of increasing the urbanization pressure. By prioritizing conservation efforts and engaging stakeholders, it is possible to ensure that continued survival of this vital ecosystem and species they support. So I can acknowledge all these institutions for working with us and helping us and supporting us to fund this project, this conference. Thank you so much.
Thank you, Ziza. We can give a better clap for 2 reasons, which I'll tell you. We are doing some kind of an exam for students who are presenting. We are doing the evaluations, and we are focusing on 5 areas. So if you are a student presenting, like Aziza, my apologies, I didn't tell you about this before. We are looking at this is a leakage on 5 things. We are looking at how your title relates to your work, how your slides are presented. We look at your diction, clarity, audibility, and confidence. We are looking at time management, and we're also looking at your quality of data analysis. This is for students only. And in this room, we have 3 of them. One has presented. We have the next one presenting, Philemon Langat, who is a student. Do we have him? Good. Thank you. I'm getting confirmation that it is negative. Yes, he's not in the room. Sorry, my apologies. Next presenter is Florence Macharia. Thank you. We don't have Florence Macharia again. My apologies. Now, whom do we have? Do we have Alois Wambua? Do you have Alois Wambua? I think I saw from Chinese Academy of Sciences. Is that correct? Yes, you should be presenting. Sorry for ambushing you. Should have come after the two, but they are not here. So I'll give you a minute to breathe, since you are a student and you'll be evaluating you. As it comes on stage, I think we need to do an exercise, maybe. Do we have someone who can guide us in doing some warm-up, just to wake us up? Any volunteer to give us a jig, just to stretch ourselves as we listen to Wambua? Anybody? Okay, in the absence of one, I think I'll request all of you to get to your feet and just stretch a little bit. This is to confirm that we are all together. I am not good in acrobatics, but we'll try. Wambua, karibu sana. It's your turn to present.
Good afternoon, everyone. Thank you for the ambush. So I'm going to present this project that I did during my master's studies at the Chinese Academy of Sciences, Kunming Institute of Zoology. I finished in 2021. It's on the comparative phylogeography of two forest specialist rodents, the Promis jacksoni and Hylomiscus endorobi, in the Kenyan highland forests, which are highly fragmented. And these are the study species that I'm going to be talking about. The Promis jacksoni, which is the African wood mouse, and the soft-furred mouse. So the underlying basics of species comparative analysis is that species that have a low population size have less genetic diversity compared to species that have high populations. And the reason why I chose to do this with rodents, they are basically good bioindicators, and they have short generation times and rapid mitochondrial DNA substitution rates. And they are hugely associated with specific habitats, and they have very limited dispersal abilities because rodents are small mammals and they cannot disperse to far places if there is no connectivity. The objectives of this study were to determine the pattern of phylogenetic variations in the two species and to unravel the effects of geographical location on the genetic diversity and to compare the patterns of genetic variation in the two. We conducted the study in Kakamega Forest, the Mau Forest, Mount Kenya, and Loita Hills. And if you look at the distribution of the forest, the ones highlighted in blue, in green, most of these forests were continuous many years ago, but now they are highly fragmented. And the Rift Valley has separated these study sites into the southwest area and the central area of Mount Kenya. And what fragmentation does into populations, it causes a genetic drift, and there is a high risk of inbreeding, because if there's no connectivity between habitats, there's no dispersal of populations. And then this results into low evolutionary potential and a very high risk of extinction of species. So we did DNA extraction and sequenced 222 samples of these rodents using 2 mitochondrial DNA and 1 nuclear DNA. And the results that came out is that there was high genetic diversity in Promys jacksoni compared to Hylomiscus endorobi. And we use haplotype networks to determine the rate of mutations. And the mutation rates, if you look at these nodes, these ones represent different populations across the study areas. And there is high mutation rates into distant populations and different localities. The same case was in Promys jacksoni. The populations also had high mutations. And using the phylogenetic trees and CYTB, there was no different, distinct population structure across the different forest fragments in Hylomiscus endorobi. When you look at this, it looks like a mix-up of colors representing different populations from Mount Kenya and the southwest. The southwest was representing Kakamega, Loita, Mau, and then there was Mount Kenya population. So there is no distinct structure from this phylogenetic tree. And then we also compared this with samples from GeneBank. For CYTB gene and Promys jacksoni, there was clear population structures between Mount Kenya and the southwest population. Using D-loop, Hylomiscus had no clear population structure again. And for D-loop Promys jacksoni, there was a clear population structure. When you look at IRBP gene, there was unclear population structure for Hylomiscus again, and also another, now this was unclear for Promys jacksoni as well. We combined the 2 mitochondrial DNA, the CYTB and the D-loop, and compared the 2 populations, and they almost look the same for the 2 species. And then this is a concatenated gene phylogeny. This means a combination of all the genes. And for the Hylomiscus endorobi, looked like there was also unclear population structure compared to the Promys jacksoni. And we did species delimitation for Promys jacksoni, which gave us 3 populations using this method called ABGD, and MTPT gave us 2 populations, showing that there are distinct populations within the same species. And conducting divergence dating, we saw that Promys jacksoni had the most recent internal divergence compared to Hylomiscus endorobi, which occurred about 3.8 million years ago. For Hylomiscus, is 6.4 million years ago. And this can be demonstrated using this diagram here. Then for the population changes, Promys jacksoni had a recent shift in population change compared to Hylomiscus endorobi. And so overall, there was a contrasting phylogenetic patterns showing unique evolutionary histories between the 2 species. And the clear distinction of between Mount Kenya and Southwestern Block for Promise Jacksoni only. And some of these populations in the same locality, like Mount Kenya, they appear genetically distant, yet they are in the same species. And the geographical patterns, we observed that the mutations rate were very high in distant locations than in close locations, and the genetic diversity was very low in close localities like Mount Kenya. Sirmon and Chogoria are very close area, but the genetic diversity was very low. And also the Great Rift Valley has separated the Kenyan highland forest into 2 blocks, which shows very distinct population differences. And in conclusion, Promise Jacksoni had a higher genetic diversity than Hylomiscus endorobi. And the Hylomiscus endorobi seems to be able to survive under continuous forest habitats, while Priomis has a high tolerance and can survive in various forest habitats, including highly disturbed ones. And lastly, the Rift Valley has resulted into isolation of populations. So we would recommend additional sampling in all the highland forests in Kenya and to conduct the phylogenetic studies. And these outcomes can be useful in defining ecological and anthropogenic barriers that interrupt gene flow across these regions. And thank you to my supervisor and Sajorek, the National Museums, and all those who supported me in my study. Thank you.
Thank you, Alois. Maybe you give him a better clap. You might have noticed a glitch in the program by calling Alois to come on stage. That was done. As you know, exams are full of surprises, and surprise is an element in examination. But he stood up to the challenge very well. So, thank you, Wambua. Our next presenter is Michelle Lawing. From the abstract, she mentioned that she's from Texas, US, and she'll be telling out about the work she has done in Kenya. So, Michelle, karibu sana. And my apologies, Michelle, for the glitch. It was to allow the judges to evaluate the students before they move to the next breakout session. So, I profusely apologize.
Okay, hello. Hi. Let's see. Thank you for being here and remaining in this session. I'm very excited to talk to you today about broadening participation in ecometrics research. I will tell you a little bit about what ecometrics is in just a bit, but this is about a group that has been it's a large international collaboration that we've been working together since about 2017, and we are Conservation Paleobiology in Africa is like the umbrella group that we're working underneath. So as we all know, we live in a warming world. This is a graph showing the measured change in temperature over the last century or so. But species have been responding to climate changes for the history, throughout the history of life on Earth. So we can learn about past responses of species to climate change by studying the past. But there are other threats to biodiversity besides the changes in climate. Human influence increases on Earth's landscape and biota. This is through human population growth. habitat destruction and fragmentation, and intensifying land use. And landscape filters through these And this means that our predictive modeling suffers in ecology and conservation. So we need strategies to integrate information about the past into ecology and conservation. So we have a lot of different types of information that we can extract from the past. There's paleoclimate models that we can learn about how climates change through time. We have phylogenetics that we've heard about in this session. We've heard some really nice phylogenetic comparative studies. We also have fossil occurrences, what fossils look like, where they were found, when they were found, and the traits that fossils hold. And so what I'm going to talk about for the next few minutes is our focus on traits and using traits to understand how species respond to changes. So I was part of this workshop that was hosted here at the museum in 2017. This is traits. It's a trait-based workshop about quantitative approaches to paleoecology, conservation, and climate change. And from this workshop, a group of us put together a grant, and we asked for money to do some work on developing taxa live in drier areas, and communities with low hypsodont taxa live in wetter areas. And we can use these traits in not only mammals, but also in a lot of other kinds of taxa. So I have another example of some traits in snakes that we've used in an ecometric model. So Jason Head has published this really nice ecometric model in 2009 on how body size, the maximum body size of a community is related to the mean annual temperature in an environment. And also we know that snake relative tail lengths at the community level are linked to the vegetation cover that the community occurs within. And what we can do with these models is look at the ecometric fit. And from looking at fit, we can calculate the anomaly of the model. And so this graph is a violin plot showing how well the ecometric models, how well the traits are fitting to the environment. And all the blue places in this plot show a really good trait environment fit. And the yellow violins show that there is a break in the trait environment relationship. And these are for 3 different traits: body mass, hypsodonty, and loaf count for mammals. And on the x-axis, we have time in millions of years. So you can see that we have some major break in trait-environment relationships occurring after about 1.8 million years. So we can also use ecometric relationships to model where we expect trait-environment relationships to break down in the future. So here's a map showing a projected climate model where we have the future ecometric relationship, whether or not it will weaken or strengthen under different climate change scenarios. And so this one is SSP 5.85. And we can look at this map and try to understand different conservation strategies to target to allow the community to maintain its trait distribution in the face of climate change and changes in land use and habitat loss. So Dr. Head gave a really nice talk yesterday about functional traits as tools for assessing ecosystem health. And today I'm going to mention the database initiative that we're working on. So this is a multi-trait, multi-taxonomic database that can incorporate traits, functional traits from any type of taxon. And the goal is to compile ecometric traits across taxa to track ecosystem change across space and time. And so we can add different kinds of traits. Like, for example, we could develop arthropod ecometrics, and we could look at body size distributions of insects or arthropods throughout space and see how it shifts through time. So we could incorporate any kind of functional traits into these models of ecometrics, which help us identify the changes in ecosystem health. So we have had some broadening participation activities. We've incorporated undergraduate research opportunities into this work. We've brought a couple of undergraduate students from Texas, from the Aggie Research Program, and we've also worked with some students here from the Prehistory Club of Kenya. And students help gather trait data and measure specimens. And we are also working with the students to develop open access materials to improve our ability to guide students through ecometric data collection protocols. We're also using these protocols in a community science framework, and we're developing a Zooniverse project so we can engage volunteers and learn about data collection and analysis in an ecometrics framework. And we also have ecometrics working groups that we host every once in a while. We are very interested to collaborate with others so we can better understand and analyze ecometrics data, and so we can gather more ecometrics data together with these ecometrics working groups. Here's a picture of one working group that we had here at the museum. So ecometrics is a promising way to integrate information about the past into understanding ecosystem health and conservation planning. And if you're interested in this work, please visit the exhibit booth that we have. And also you can email conspaleoafrica@gmail.com. Thank you.
Good. Thank you, Michelle. You've been gracious in 2 ways. One, you have saved us 1 minute. And 2, you had so many photos in your presentation. Thank you. Now, the sun is coming down. And so is our presentations. We have 3 more. We need to hear from I'll mention their names. We need to hear from Gilbey Obuga, Lawrence Wagura, and Titus Iboma. And before that, I'll make an announcement. That if you registered for the cocktail event happening from 6 p.m. today, we'll be meeting outside the gallery, the main gallery, from 6 p.m. if you registered. So we have already heard from Alois, the ICT, please kindly, we have heard from Alois. So now we need to hear from Gilbey. Do we have Gilbey Obuga? Then after Gilbey, we'll hear from Lawrence and Titus in that order. Thank you. Welcome, Obuga.
Good evening. I'm Gilbey Obunga. I work for Nature Kenya, and I'm stationed at Taita. That is where my workstation is. I'm here by you to present part of my research thesis on habitat suitability, modeling to improve conservation status of 2 critically endangered endemic Afro-montane forest birds species in Taita Hills, Kenya. These 2 species, the Taita pales, the top one, and the Taita thrush, they have been classified by the International Union for Conservation of Nature as pales being critically endangered and thrush being endangered. The 2 are on the verge of going extinct owing to increased habitat loss and fragmentation, degradation, and climate change impact. Therefore, there is need for exploring their habitat suitability to ensure that they continue to persist. The aim of this study was to determine the current spatial distribution and the key geographical explanatory factors and conditions that affect Taita pales and Taita thrush distribution in Gangao and Buria forest. Both are found in Taita Hills using a maximum entropy model. To achieve this, we have 2 specific objectives. One was to examine which and how ecographical factors influence the habitat stability of Taita pales and Taita thrush. And the second one was to create maps of predicted occurrences of the 2 studied birds in the respective forest fragments. Our research was guided by these 3 research questions. One, which eco-geographical factors and how do they influence the habitat stability of Taita pales and Taita thrush in Gangao and Buria forest? The second question was, which parts of Gangao and Buria forest are suitable for the activities of the Taita pales and Taita thrush? And the third one, what is the percentage of suitable habitat currently employed by Taita pales in Gangao and Buria forest and Taita thrush in Gangao forest? There is my study area. The 2 forests, this is the Gangao, and here we have the Buria, located within Dawida Hills of the Taita Hills. On methodology, the species occurrences were collected in June and July 2019 and 2020 using standardized point counts techniques where we developed a grid, 100 by 100, which was used for data collection. At each sampling point, coordinates of the location where the bird was sighted was recorded using a Garmin eTrex 30 handheld GPS receiver. Data collection was done early in the morning and late in the afternoon when the birds are active, where a total of 23 occurrence records for Taita Pallis and 30 for Taita Thrush were collected in Ngangao Forest, and 21 occurrence points for Taita Pallis were collected in Vuria. And these data were used for modeling. We used 5 different data sets. The first one was data related to LiDAR, airborne LiDAR, that is the canopy height and relief, point cloud penetration, and percentage of the fourth returns. The second was topographical related data, that is the elevation, slope, and aspect, topographical wetness index, and profile curvature. The third was a variable which describes forest vegetation interpreted from airborne hyperspectral remote sensing data. And the fourth one, landscape variable based on Euclidean distance calculations. And finally, climate-based variable, that is the annual mean temperature and annual mean relative humidity. In calibrating the model, like I said initially, we used maximum entropy version 3.33e. These are a machine learning algorithm where 75% of the occurrence data were used to train the model and 25% for model performance determination. We also did 10 random partitionings to assess the average behavior of algorithm. And this one also allowed for statistical testing of observed differences in performance. Under the model evaluation, we used area under the receiver operating characteristics curve, that is AUC. And the value ranges from 0 to 1, where 0.5 indicates that a model didn't perform better than the random, whereas 1 indicates the perfect discrimination. In this modeling, a model with the highest test area and curve values were considered the best performer. Previously, before that, we developed 3 models. The first model was composed of independent variables. That is, all the variables were run independently of one another, and the top 5 performance were used in the first model, and then the second model had all the variables, and then the third model composed of top 3 variables with the highest area under curve values per each category. That one for the Ngangao modeling and Vuria. That one was for Ngangao modeling for Taita Pallis and Taita Thrush, whereas in Vuria we only used 2 models, where the first model entailed a top 5 variables per category, and then the second model had all the parameters. We generated habitat suitability maps based on the prediction models, and these maps were reclassified into 4 broad classes. The first class entailed unsuitable, that was between 0 to 0.2 probability values. Then 0.2 to 0.4 was reclassified as a moderate suitable site. And then we had the high suitability areas 0.6 to 1. Based on the findings of these results, all the 3 models performed well, owing to the area under curve values, which were found to be above 0.7. There was also very low overfit in the prediction results, as indicated by small differences in the test and training values. So there are the values related to the training. For the test data, I indicated initially that we use the test data to determine the performance. We are seeing that all the models posted at least 98%, no, at least 78% accuracy. So we have, for example, the models in Ngangao and Vuria had, the Taita Palace model had 80% accuracy and 89% accuracy in Vuria. For the Taita Thrush in Ngangao, the model performed at 78%. So here are the maps of the maps showing areas within Ngangao which were predicted to be suitable for the two birds. The first one on the this one indicates areas which are highly suitable for Taita apalis. The red indicates highly suitable areas, whereas the yellow indicates moderately suitable. The white area, those are the areas which were masked from the analysis, since they are the areas which are covered by rocks, and these species are known not to be found on these rocky areas. This was for Taita apalis. Then on the right, we have the distribution map for Taita thrush, indicating areas which were predicted to be highly suitable and areas which were predicted to be unsuitable for the species. Then we have Taita apalis presence in Vuria. Again, it is indicated. We have this, the southeastern part, which is this area. This indicates Vuria forest. And then we have the northern patch, which is Msidunyi. Then we also examined the response curves, whose contribution to the model were slightly more than 10%. And based on the variable curves, we were able to determine which parameters which affect the distribution of these species, of the two species. For example, we found out that the LiDAR-based canopy height range and elevation greatly influence Taita apalis distribution in Ngangao forest, with areas of high elevation, that is between 1620 to 1720 meters above the sea levels, were favored, including areas with open mid-stories. Then for Taita thrush in Ngangao, elevation, slope, topographical wetness index were the major determinants of the thrush occurrence in Ngangao, where the gentle sloping areas with moderately dry surfaces, and again within 1620 to 1730 meters above sea level, were the most preferred. Coming to Taita apalis in Vuria, mean annual temperature, Euclidean distance to the forest edge, slope, and land cover type greatly influenced its distribution, with gentle sloping areas within the forest interior composed of indigenous vegetation were the most preferred sections. So we also did analysis into quantifying the area covered, the areas which are categorized as highly suitable, moderately suitable. And based on that, we concluded, we realized that less than 7% of the two forests are considered to be highly suitable for the activities of the two species. And what does this one tell us? It tells us that the efforts towards ensuring that these species don't go extinct should be directed towards preventing further habitat loss and disturbances, restoring habitat quality, and increasing habitat connectivity between the two forests. Thank you very much.
Thank you. Obuga. Sorry, time had to be observed, and we didn't see one or two slides, but we got to do that. Anyway, do we have Lawrence in the house? Good. Lawrence, your work seems similar to what we have seen, so we could combine probably, and you tell us more about your work. So kindly join me on the stage. Maybe a clap as he comes to tell us more about the birds in... Okay, I'll not preempt. So, kindly.
So, good evening. My name is Wagura Lawrence. I will be presenting on more or less the same. On the same species. One of the species my predecessor just presented on, and more or less the same area. So, but I'll focus on the population traits of the species. I think it was somewhere on the list, the long list. Can I find? Apparently, I cannot find it. It's yeah. There you go. So good. I'll present on population traits of Taita pallis. It's a critically endangered species, which is endemic to the Taita Hills. And it's a work that it's a long-term surveys that we've been conducting together with the team, as in the list. So yeah, a lot of presentation on Taita Hills have been done today and even yesterday. So I will just highlight on the area for the benefit of those perhaps who are not here or what was not mentioned before. So Taita Hills is part of the Eastern Arc Mountains, which is comprised of 13 mountain blocks. And the Taita Hills is the only block in Kenyan side. The rest are in Tanzania. See the way they run from southern Tanzania all the way to southeast Kenya. So Taita Hills is the most fragmented block among Eastern Arc blocks. But surprisingly, and I can mention that it's the most also degraded one, but surprisingly continue to support a high number of species which are unique to the area. There's a lot of endemic species. In fact, per unit area, it has the highest endemism among the Eastern Arc Mountain blocks. Okay. So that's a study species. It's a small warbler, which is morphologically very similar, male and female. Apart from a slide, there's a male. See it's more darker on the throat than the female, and of course the call is different. They prefer, like I said, it's endemic to the hills, and the preference habitat is in the forest gap and along forest edge. We've been conducting all along quite a good number of research there, including the demographic study, which has been the most main focus area. Using various methods, such as point counts, ringing. We've done studies on breeding ecology of the species. We've done radio tracking and the rest. But today,
in this presentation, I would like to focus on point cloud surveys. We've been conducting this using a preset grid, permanent grids. That's points in the entire forest spaced 100 meters apart. So we've done analysis of this point count over, on the data that have been consistently collected. The decline continues. In one of the fragments called Yale, we only have a pair existing there, so it cannot be considered as a viable population. Some of those major drivers for this population decline that we have identified is quite several, but the key one, and we continue in these investigations, we have witnessed high predation rates. And as you can see, all these are predators of the species. There's cyke monkeys, we have dormice, we have goshawks, we even have galagos predating on these species. Some of these causes are not known. You only find the species, an individual dead in the nest. So this is a major, is one of the major cause of this decline, and it's serious because they are all natural predators. Another driver is low nesting success, and in some extent, post-fledging survival rates. We have, because we have also conducted a lot of work on nesting, breeding ecology of the species. So we have done a lot of nesting surveys, and we have noticed that most of the predation is happening between the egg to the pulling stage. And if I am to explain in an easy way, if you look at this chart here, you find like in one of the forests, that is in 2020 and 2021, we have had, like in one of those small fragments, we have had 8 territories, out of which 6 were occupied. 4 pairs had nest, and they did 15 attempts, because once the breeding fails, once the nesting fails, they repeat. They tend to repeat until they succeed or they fail completely, and the season get out. So you can see 15 attempts and 14 failed, and only 2 fledglings, and only 1 survived. Similar in Gangao, 15 and 12, and then they did, out of these 12, they did 61 attempts. All of them failed. Whole entire season, breeding season, they all failed. So, and the same trade in Vuria. And somehow those that survives, because we tend to monitor these nests until part of, once they post-fledging, we do a bit of follow-up, but these need to be extended to, because we don't see these adults. We don't know where these are, we don't know whether they get to the adult stage. We only follow to a certain stage, up to perhaps immature stage, and then after that, we don't know what happened. So again, there's a limitation of habitat and serious fragmentation. One of these forest fragments, like in Nyale, it's in the right altitude. We have had a single species surviving here, a single pair surviving here for I think 6 to 7 years now, but the population doesn't increase. And one of the major reasons is that all these sites highlighted here are plantation, and only a small portion of indigenous forest. And this is not enough. Perhaps that's why the population doesn't increase. So there's a challenge in the of the habitats. And also you can also notice how fragmented these forest fragments are. They are quite far apart from each other. That could also be a problem. We also think that climate change is involved in this, and the reason why, but this is just what we think. Perhaps it needs some work to prove this. But the local extinction, as you see from here, it's quite related with the altitude. So in Vuria, where we have is the highest point in the Taita Hills, at 2061. Still have the number. The population, we can say, is relatively stable. Msudunya as well. Yale as well in the right altitude, but then the habitat is quite tiny. In Gangao, that's where the altitudes are going down. You see, you can notice the sharp decline. Bololo, extinction already. You see the altitude. And Fururu and Chawia, that's where the extinction has happened. So that could depict. Is the time already? Okay, 1 minute. So we don't want to do. We just don't want to do research of these species until we record, global extinction of the species, and that's given the number that we just highlighted, 200 individuals, making global population is not a stable population. That's not sustainable. So we are trying to do some conservation work, even as we do continue with our research. We are somehow trying to expand the natural habitats through clearing of exotic species and replacing them with indigenous species. We are working with the community who are really helping in this. We trying to map those areas that are high priority, and we working with closely with Nature Kenya, who have really done a lot of work in purchasing and even leasing those areas that have been mapped as high priority. We are also trying to do some connectivity work to, make sure that there are corridors that can help this subpopulation or those subpopulation interact. So this is just food for thought. It was supposed to be a video just for you to hear the call, and perhaps might provoke the thought of how long will we hear the species singing, or are we really doing enough to make sure that it's sing forever? Because it's a species that for sure it might become extinct. Media, if you can play the core, or if it's not possible, then it's fine. So, and is it possible to play? No? Okay. So it's fine. So thank you so much for your patience and listening.
Thank you, Lawrence. That was powerful. I would have wished to listen to the bird sing. I'll see you. You may attend. See you. Yeah, thank you. We are doing our last presenter for the day from Titus Iboma. Titus, please welcome to the stage and take us through the last part of the day. Then from there we can do a few questions, then we wrap it up. Thank you.
Good evening. I'll be leading you through my second discussion. This is not the one, please. Okay. Okay. My name is Titus Imboma, taking you into my second discussion today. And in the morning we looked at tea and birds, but we want to look at some discussion on birds and urban areas, or we call it green spaces in Nairobi. And we are going to base this on the Green Museum as a green space, one of the important green spaces in Nairobi that is attracting migratory birds around. So just as they come through, yes, thank you very much. So the use of green space by migratory passerines in Nairobi City. The passerines are small birds that can perch compared to huge birds. So we want to look at those ones that really have used the museum's botanical gardens as one of the key wintering sites. Next slide. Next slide. So we find that every year, millions of migrating birds leave Europe to Africa, and some will leave North America to South America because of the winter seasons, to what we call from breeding to wintering sites. Africa becomes one of the very important wintering birds of winter area for birds from Europe and Asia. And we find that about 170 birds are what you call palearctic migrants. Those are birds moving from probably Europe and Asia to Africa every year. In Kenya, we have got about 170 species. And we also have what we call afrotropical migrants, which are 60 species of birds. Next slide. Okay, to look at the birds in the museums, this is the botanical garden. We used mist nets to capture the birds, and we used nets of about 56 meters long within the botanical gardens, and birds were captured from 6 o'clock in the morning to 11, and these are done every week on Tuesdays. Next. So we found that we studied palearctic migratory birds within the museums. At the headquarters, the botanical gardens comprises of a variety of habitats, including riverine habitats along Nairobi River, wooded vegetation, enriched with indigenous trees. Enriched means they have been planted by the botanical garden, adding them to make them a better place. And also succulent gardens. They support a lot of biodiversity, so especially of birds. Now, interlinked with the Mijuki Park, now that we found that we've had a very nice environment for not only just wildlife, but also for the birds to come and for the people to come and enjoy. So, so the results, our results show that, and this is 10 years, a span of 10 years that we've captured these birds. And our research shows that a total of 330 birds of 17 species have been captured here in the museums in a span of 10 years. And we found that the most common or abundant species within our nets in the museums are what we call the willow warblers. This is the smallest migratory birds of about 9 grams, which takes 25% of those 330 birds captured. So this is followed by also another smaller species, which we call blackcap. The blackcap is the species that was on my first slide, which takes about 17% of the birds captured. So we found that if we're looking at this, we are talking of about a total of 17 species. That's quite a number of birds moving from Europe and utilizing our habitats here. And they range from different species, like what I mentioned, the willow warbler. Sorry. The willow warbler leading the number to about this, and this to the lowest numbers of things like the great reed warblers. So in some, we have got quite a bigger number of these representatives of this species, and some is quite low. These low ones means these are the birds that have come in just probably in 1 year. Others really come every year, like these ones, the blackcap, and also the willow warblers. So it's a nice representative of what we get in the museums as far as migratory birds are concerned. So we looked at timing. We realized that some of these birds come here for specific time. They're not just here throughout. And different species have shown that they are here specifically for some different times of the year. So we divided this into, from September to December, some birds will arrive as early as September from Europe, when the winter starts, and others will go and then they will go up to May, when they go back to Europe. That's when the summer is beginning and the birds are going back home to breed, because this is only the wintering place for them. They will breed back in Europe. So some of these species are very interesting. For example, the willow warbler, which we get the biggest number, will go on about and reach only April. Then from May, you can't see this species. Some species like the reed warblers will arrive in October and reach April. So most of the birds are reaching April. But this is a very interesting one that comes in and goes all the way to the May. And May is when almost all the birds have left from Kenya, from different areas, have gone back to Europe. So we might find that these birds will stay here for quite some time. Other birds will come in, especially when you look at the cardinal warbler. It's just throughout the time. So it is surviving in the museum all this time of arrival in September until it leaves back in May. Very interesting. However, other species are just here this time of the year, which means they have either come from other places, but they don't pass through museums. They come to museums to fuel, put on fats to prepare to fly back home, and then they leave. But we don't see them when other birds are coming in, like say here, they're not here. So they are only here in the museum during this time. Others, however, are just here during their arrivals, and they disappear. Migration, as you understand, when they leave from Europe to Africa, they'll go all the way to South Africa. So some birds will go to South Africa and spend most of their winter there, and then they go back to Europe. So the data is showing us that some birds will really stay here. Some birds will make only this place as a stopover to fuel to go back home. Others will just come and then stay for some time and continue to South Africa until the South African region. So in a conclusion, we found that 17 species, as you've seen, find their way, that among the 170 species of palearctic migratory species, find their way on the museum gardens. And this is because of the how well the garden has been managed, and we're seeing a lot of improvement every time. So they winter here and take most of their wintering time here, while others will just pass through, and others will come when they only want to go back to Europe. So migratory species are protected under the CMS, and Kenya is signatory. And when we see such habitat playing a very important role, we are encouraged to give it an applaud for conservation, especially now that Michuki Park is connected to Nairobi Botanical, to Museum Botanical. That's one of the way the migratory species can be supported. Yeah, we find that species richness and diversity play an important role, even in ecotourism. We've seen many people coming here with binoculars just to look for birds at the Michuki Park. Yeah, so the museum garden becomes a very important site for that. So in recommendation, we need to enrich the available screen spaces with indigenous trees, make them available so that they can support biodiversity. It's not only just birds, but I know many other things will be here very interesting. So there's need to conserve and manage the riverine habitats within the green spaces. We've realized that for some time, Nairobi River has been very bad, especially this year in terms of sometimes it looks like sewage and so on. And some of these places, it's not only migratory species, but also some unique riverine ducks play a very important role as their site here in the museum, especially those that move along the clean waters along the highland places of Nairobi. There's also need to document availability of biodiversity within our green spaces. If we had not done this, we wouldn't know how much species of migratory species are using. And I must applaud the botany or the herbarium for having labeled all the birds, all the plants, not birds, within the gardens, and it's very nice. This is improving our awareness about the museum. Thank you very much, and for the entire green spaces in Nairobi.
Thank you, Titus. When the lights go off, it means that we are coming to the end of our session this afternoon, which has gone to the evening. And I think we had an agreement that we'll take questions and answers. Could we again agree how many minutes we need for that? 5, 10, or less, or thereabouts? Do you have anybody with a question? Either recorded, noted down, or oral? We're going to give let's say 5 minutes. Is that fair? Okay, I see some nods. I do agree. 5 minutes of questions. And I have my questions, and I'm looking at some of the presenters. I can't see them in the hall, so I'll follow up with them. Are we in the same predicament? Maybe not. Good. Yeah, Christine, we have a question on the middle row from Dr. Wanja, kindly.
Thank you. Is it? Yeah, thank you. I just wanted to compliment or congratulate the BART team that is looking at the green space in especially the Botanical Garden and the Michuki Park of Nairobi. And this is simply because some of the work we have done on pollinators in the area, we recognized the importance of this green space. And we have realized that Nairobi is actually expanding exponentially. So we really do need this space. And those of you who are in the plant group, I think we may need to link up so that we can relate the pollinators with, especially the herbs and the lower plants that we are not very comfortable giving names. So there's a lot of work that can be done And I urge all other institutions to ensure that they have some green spaces that even their students can work at. Thank you.
Thank you, Dr. Wanchet. That's a very nice message, take-home message. In your institution, if you have a green space, then you expect increased biodiversity, both in abundance and diversity. We have a question, Christine, on the left side. We have a gentleman just next to Dr. Jen. I'm getting to know all of you. By the time we have finished this conference, I'll have known all of your names. And I think that's a good thing.
I have
Thank
a
you.
question for Jenna. And I wanted to look at diversity because when they made the presentation, there was no significance difference in terms of diversity. But would that diversity not be different because everything is cramped together? Because if you are looking at indicators, there will be indicators that will increase because of change, and there are indicators that will decrease because of change. So if you cramp everything together, don't you negate everything such that it doesn't? And then I had a question also for Lawrence here. So if you are looking at 200 species, is it a problem of habitat modification, or are you looking at inbreeding? Because maybe if you're looking at 200 individuals, I'm assuming there is inbreeding. So is it habitat modification, or is it actually inbreeding that is happening, such that the population will not increase?
And before you answer, kindly, we need to put on record the one asking the question. You didn't tell us your name. Kindly.
Dr. Mwangi, International Crane Foundation.
Thank you, Dr. Mwangi. So, Dr. Jen.
Yeah, thank you so much. Yeah, we took note of that. And the space we are working on, Ewansonarok, is like one space. It's a 17-kilometer stretch. And our plots were basically on the same, what do I say? It's like working in the museum compound from one corner to the other. And we took note of that. And that's why the diversity there is no significant difference in diversity. Again, you know, insects migrate, they fly from one plot to the other. That still also reduces the diversity. But our main objective was to first have the baseline study on that, and then we see how going forward we can build on it as a monitoring aspect. Yeah.
Thank you for the superb answer. Lawrence.
Okay. Thank you, Dr. Mwangi, for that question. We haven't looked at inbreeding yet. It's an area that we want to go to. But then, it could be that there's some genetic erosion, but it's an area that we will venture. But still, looking at some of the forest fragments, like in Vuria, we've had the population is doing well, and it's a tiny forest compared to the other forest fragments where the species have become extinct. Bololo forest fragment, it's like 10 times the size of Vuria, and the species have become extinct. So that could now start ruling out the issue of inbreeding. So, but it's something that ought to be looked at.
Yes, Dr. Jen, you have a question?
No, mine is not a question, but I want to appreciate the work of Dr. Iboma and the work of Dr. Mwachara and the team that did the field guidebook. And I think it will be good for us to work as a team, because when I think of, maybe if you have to think of our institution having a green space and the surrounding green spaces, I think of like how we can team up together to come up with like income generating activity for our institution,
work because like it's an ecotourism thing for the birds, for plants. And I've been saying that I've been doing documentary in Gong, you know, the video documentary. So, I mean, it's just a general comment. It's only that I really want to appreciate the work we are doing, but maybe if we work together, we can promote our institution and the Nairobi city as a metropolitan. And now that they are also thinking of the Nairobi River Basin Commission, I mean, it's amazing to see that we are working alone, but together I think it can be a better output. I was really delighted to see the output from Dr. Mwachara and team, and now Iboma. And I wanted also to say I've been doing documentary with some funding from JRS. And I mean, you can imagine if we do the documentary and you are doing this. Thank you so much. It was just a comment.
Thank you. We had an agreement of 5 minutes, and you nodded your heads. Now we are breaking that agreement. In the interest of science, do you break the agreement? Yes, we do. Gilbey.
Okay, mine is just a comment on the two species we've just presented on. I think a lot has been done, and in terms of monitoring and bits of research, but still the population is going down more so for the Taita apalis. And I do think that there is need for the next stage or the next step that would entail probably looking into possibilities of undertaking in captive breeding. It may be a bit challenging owing to the size of the bird, but we should be seriously thinking about creating more habitat, expanding the existing habitat, and then thinking of reproducing other individuals, and then probably doing bits of translocation. Like there was one which was done for the thrush from Bololo to Chavia, and the species are still there. So we should critically think about trying both the translocation and captive breeding. Thank you.
Welcome. Lawrence, do you have another question? Good. And as I do that, I think just to refresh our memories, we have this afternoon handed out reptiles by Dr. Malonza. We have heard about invertebrates by Maurice Mutua. We have heard about birds, maybe to Flamingo, maybe to Gong Hills, Taita Hills. We saw some work done by Alois. And we also had some work done by Michelle. So it's been also, not forgetting James Odanga on the Bactocera, on avocado. In Taita Hills, maybe next time you look at FCM, which is another very serious pest, which is the nice market for avocado as well. So we have had a very rich afternoon, haven't we? Yes, we have had. So I've had the honor and the privilege of leading you through the session. I'm most humbled for your time and for your attention, and I look forward to the rest of the days. I need to thank you for your patience and for the science. So may science live forever. I want to bring this session to a close. If in agreement, are we? Good. So we agree to end it there. From here, we'll have a good time. We'll have the cocktail party beginning at 6. Which I think is almost there. We are at almost 6 p.m. So from here we meet outside the gallery, the main gallery. We'll do 2 hours of some social interactions. We have done the scientific part. Then we call it a day. Thank you very much. I've had a good time as well. Asante sana. So today's session is officially over. Thank you.